Okay, it's connected. Okay, so okay, people are starting to, I see.
Okay.
I see attendees coming in. Okay, welcome to everybody. We had a few technical issues. So, and let me start the live streaming in the meanwhile. Okay, yeah. I guess we had more time for people to connect. So let's see. Sorry, everybody, we were a bit delayed because we had technical issues. Welcome. Give us 2 minutes so we settle with the connection and we will start the lecture. Okay, so we are going live on Facebook too. Okay, streaming live in 1, 2, 3. Let's see, people have connected. Okay, one moment. Okay, we are live on Facebook now. Excellent. Okay, so thank you for everybody who had the patience to wait through the connection difficulties. I guess that's what happens when you have to deal with technology sometimes. So it's with great pleasure to welcome you in our 6th season of the Cyprus Biological Society lectures. And we are kickstarting this season with Dr. Chiara Francavilla from the Denmark Technical University and also from the University of Manchester, as she holds dual positions. And Chiara is going to take us through different approaches to understanding cancer signaling. So before I give her the floor, I will just go through a very brief introduction of her bio, and then she's all yours. So Chiara obtained her master's degree in industrial biotechnology from the University of Milano Bicocca, Milan in Italy. And then she did her PhD in molecular medicine at the Institute of Molecular Oncology and the European Institute of Oncology. She was a Marie Curie intra-European and an EMBO long-term postdoctoral fellowship fellow at the Novo Nordisk Foundation Center for Protein Research at the University of Copenhagen until 2012. And then she was a senior postdoc until 2016. She then started her research group at the University of Manchester, where she is currently an honorary research fellow. Then very recently, in July 2023, she moved to Denmark as an associate professor at the Danish Technical University in the Department of Bioengineering and Biomedicine, where she currently studies, as we mentioned, the principles and the regulation of signaling architecture in a variety of models using a multidisciplinary approach. So I'm very looking forward to hear your talk, Chiara, and thank you for accepting the invitation and for kickstarting our lectures. Thank you, and the floor is yours.
Great, thank you really a lot for the introduction. The kind of introduction is very exciting to be here and give this online seminar from my office in Copenhagen, in Denmark. And what I would like to start with is actually to guide you through the title of my presentation, which at the moment is Functional Cancer Proteomics to Study Signaling Architecture in Breast Cancer Models. And these are a lot of difficult words, and I want to start with what I mean with signaling architecture. Okay, so first of all, when you think about a cell, a mammalian cell that needs to survive, you think about the fact that the cells need to balance different responses. So the cells can grow, die, or move to survive. And this happens during development and also in our adult life. And the key point here is that cells are exposed to different perturbations from the environment. For example, we can think about a different amount of nutrients in the environment, the presence of chemicals, including small molecules like growth factors, and even mechanical forces that from the extracellular matrix. And all these factors affect cell behavior and needs to be translated as signal to regulate the balance of different responses. And how do the cells do that? One way in which they can do that is through members of the plasma membrane. In particular, my group study receptor tyrosine kinases, which at the end regulate different internal cellular process like metabolism or gene transcription to fine-tune the balance between different cellular responses. And how do they do this mechanically is by activating signaling pathways. So this is why signaling is actually the keywords in this talk and in the work that my team is doing. Signaling, therefore, is important during development and for adult cell homeostasis, but an important point for this presentation is that this regulation of signaling underlines diseases like cancer, breast cancer in particular in this case, diabetes, genetic diseases, and also aging. And that is the reason why signaling molecules are actually the main drug targets. And this is why also we need to know more in spite of decades of research on signaling in order to find new drug targets. The approach that my team has been using in the last few years to study more in-depth signaling is actually to what we call functional proteomics approach. So we always start from different experimental conditions to compare. We can use normal cell lines, 3D cell culture, primary cells, ex vivo models, and even human biopsies to get samples to compare upon perturbations. And then we can quantify changes in these samples by different methods. And we focus mainly on changes in proteins by extracting proteins and analyzing, digesting them into peptides. And at this point, we can either look at change in protein abundance based on perturbations or change in post-translational modifications, in particular phosphorylation. And to do that, we use mass spectrometry analysis. So we digest proteins and analyze the peptides in the mass spectrometer. And we do a lot of bioinformatics and system biology approaches with the idea to narrow down candidates for validation. So then we can go back to the lab, use a little bit more traditional techniques to validate our results, and eventually ask novel questions and go back to our approach. So this is what is going to be presented in my talk today. And in particular, I would like you to guide through two different questions. The first one is, how are growth factors responses regulated in breast cancer cells? Most of this work is published. At the end, I can also highlight some unpublished interesting data. And then the second part, which is completely novel, not published yet, but ready to be submitted, is which is the signaling signature underlying breast cancer metastasis. So let's start with the first one. And I go back to this cartoon showing how cell signaling can regulate the balance between long-term responses. In my team, we use the following model to answer to some of these questions about signaling regulation, which is cells expressing fibroblast growth factor receptor 2b, which is particularly expressed on epithelial cells, and that can be bind to and activated by 4 different growth factors in the extracellular environment, FGFR1, 3, 7, and 10. And this model system is important during development, for example, for mammary gland formation and lung formation during development, homeostasis, for example, for healthy breast functions, but also when there is a dysregulation, including SNPs in the gene of FGF receptor 2b or FGF10, many diseases can arise, genetic diseases, but also breast cancer. That is the topic then of this talk. Over the years, my team and I have been elucidating different ways in which signaling can be regulated. For example, we try to understand how different ligands binding the same FGF receptor 2b can generate different signaling pathways to then regulate responses, what we call structural aspect of signaling regulation. We also try to understand from a proteomics and phosphoproteomics point of view how the presence of scaffold protein in the cytoplasm affect signaling regulation upon perturbation, so the genetic aspect of signaling regulation, and also how much the signal duration affect, so events happening minutes or in hours, how much they affect at the end the balance of cellular responses. What I'm going to focus on actually in this talk is the spatial regulation of signaling, and in particular, as you can see here in this cartoon, when a growth factor binds the RTK, you can see an RTK here in green by a confocal image, and the cell nuclei are in blue in DAPI. So when a growth factors bind the RTKs, the complex is actually internalized over time in vesicles, shown here by green dots in the cytoplasm, the so-called early endosomes, from which there are two possible fates. Either the complex goes to late endosomes for degradation, and then you don't see any green signal anymore over time, or from early endosomes, the complex can go to recycling endosomes and then back to the cell surface, meaning that over time, like here 90 minutes, you will see again the receptor at the cell surface. And the key point here is that there are data showing the RTK signaling started when the receptor is at the cell surface, but also when it is in the early endosomes, or even some signaling pathways can be activated from the late endosomes, like the mTOR pathways responding to change in nutrients conditions. What was not known since a few years ago was the role of recycling endosomes in the spatiotemporal regulation of signaling outputs. So the idea of studying recycling-dependent signaling actually came back from my postdoc time in Copenhagen, where we found out that when epithelial cells overexpressing FGF receptor 2b were stimulated by FGF10, one of the ligands for FGF receptor 2b, a ternary complex could be formed by the receptor, PI3 kinase, and an adapter protein called SH3 binding protein 4. This complex couldn't be formed in the cell stimulated with FGF7 for one molecular reason, shown by phosphoproteomics. The fact that the cytoplasmic tail of FGF receptor 2b contains a tyrosine 734 phosphorylation only upon FGF10, but not FGF7 stimulation. So this ternary complex actually was important for FGF10 dependent FGF receptor 2b recycling, sustained signaling activation, and a pro-migratory phenotype. In contrast, FGF7 stimulated cells induce transient signaling activation, FGF receptor 2b degradation, and then a pro-mitogenic phenotype. So we could conclude that different stimuli activating the same receptor via different trafficking routes regulate different cellular outputs. At the beginning of my adventure in Manchester, actually, I started wondering whether this was a little bit of a more general concept, and to prove, and also a little bit important, if it was important physiologically speaking. So we try to find another model to test the same idea, and we decide to use 6 breast cancer cell lines of known molecular characteristics which resemble the phenotype of breast cancer patient, so the characteristic of breast cancer patients. And we develop what we call the trafficking phosphoproteomics approaches. So for each cell lines, we stimulate the cell lines with FGF10 or FGF7, exactly like I just described before, what we call recycling ligand-dependent signaling that we analyze by changes in phosphoproteomics in all these conditions. And we combine this analysis of the phosphoproteome with the recycling RTK-dependent signaling signature, still analyzed by phosphoproteomics, where we actually stimulate a couple of these cell lines expressing 3 different isoforms of FGF receptor with 3 different ligands known to induce recycling only of this FGF receptor isoform upon internalization. And finally, the third approach was recycling adapters dependent signaling, where we actually depleted one of the breast cancer cell lines with from 2 proteins. SH3 binding protein 4, which I just mentioned before, is a recycling adapter for FGF receptor 2b recycling, and RCP called also RAB11 FIP1, which we also demonstrated being important for the recycling of another receptor tyrosine kinases, epidermal growth factor receptor. Both SH3 binding protein 4 and RAB11 FIP1 are actually known to be overexpressed in breast cancer patients. So this approach, and as I told you before, FGF receptor in general is associated when dysregulated to breast cancer. So somehow we wanted to know if there was some new signaling signature downstream this receptor using this approach. And what we did actually after a lot of phosphoproteomics and data analysis is actually summarized here. So we took all the phosphorylated proteins that were equally dysregulated in all the three approaches, and we found out that there were different functionality among these proteins. What we catch actually our attention was that we found that dysregulation in the phosphorylation of epidermal growth factor receptor. And then we were really surprised because I just told you that we wanted to know what was the link between FGF receptor recycling and signaling. And the proteins that seems to regulate this was actually another RTK, epidermal growth factor receptor. And in particular, there was a dysregulation of a threonine 693 on EGF receptor. We validated these findings by Western blot. For example, here you see one of the breast cancer cell lines we use in our screening, stimulated with FGF7, 10, or the two canonical ligands for EGF receptor. And you see that only FGF10, but not FGF7, could activate the phosphorylation of threonine 693 in these samples at a similar level of the canonical EGF receptor ligands. Leading us to the conclusion that this was not an artifact. This event was not an artifact of mass spec, but it seems to be biochemically important. And we also verified this growing in cell culture as breast cancer organoids from a triple negative breast cancer patient in collaboration with the Manchester Breast Cancer Center and Robert Clark's lab. And these organoids to grow in culture needs both FGF10 and 7, but also EGF. So after a while in culture, we removed the FGF10 and 7, EGF, leaving FGF10 and 7, and still you could see the phosphorylation of threonine 693 EGF receptor, as in the wild type normal conditions. So we wanted to validate further this finding, and actually I summarize here some of the published work here in this model. So when cells are stimulated by FGF10, FGF receptor 2b binds to SA3 binding protein 4. It enters into recycling endosomes between 0 and 20 minutes upon stimulation, and then thanks to the activation of FGF receptor and the phosphorylation, so the activation of MAP kinase like ERK1 and 2, but not P38, FGF receptor phosphorylated threonine 693 on EGF receptor between 20 and 40 minutes upon stimulation. At this point, a complex is formed on the recycling endosomes between FGF receptor 2b, EGF receptor, and Rab11 FIP1. And this complex is important to allow the recycling of FGF receptor 2b to the cell surface between 40 and 60 minutes upon stimulation. What happened when it's not possible to phosphorylate threonine 693 anymore? So we use a mutant in this case. So we found out that FGF receptor 2b can still be in the recycling endosomes between 0 and 20 minutes upon stimulation. But then, since a complex between FGF receptor, EGF receptor, and Rab11 FIP1 could not be formed anymore, the recycling of FGF receptor 2b back to the cell surface happened earlier, so already between 20 and 40 minutes. So we concluded that EGF receptor threonine 693 phosphorylation upon FGF10 stimulation of cells, or breast cancer cells, actually regulates the timing of FGF receptor 2b recycling. We were quite intrigued by this fact, and as I told you before, recycling could affect signaling. So we decided to go further and set up a new phosphoproteomics experiment in which we compare change in the phosphoproteome of a breast cancer cell line expressing wild-type EGF receptor or the mutant version of EGF receptor upon stimulation with FGF10 for different time points. And we found a cluster of proteins here which could not be phosphorylated anymore in the presence of the mutant for the 2 time points that we use in the experiment. And among these proteins, gene ontology enrichment analysis showed many proteins involved in the regulation of cell cycle. So we did some experiment, and we found out in a panel of breast cancer cell lines the following. that while FGF10 can induce the entry in the cell cycle tested by DU incorporation in wild-type cells, in EGF receptor wild-type expressing cells, in the presence of threonine 693A EGF receptor mutant, this FGF10-induced proliferation, didn't occur anymore, or it was really similar to control cells. So we added a new piece of information to this cartoon, concluding that while the data that I show you published in 2013, so about epithelial cell overexpressing FGF receptor, point to FGF10 and FGF receptor to be recycling as regulators of motility, of cell motility phenotype in cells, it is the permanence in the recycling endosomes for the right amount of time that allows cells to coordinate cell motility with the entry in the cell cycle through phosphorylation of the activation site of one of the cell cycle regulator called CDK1. Is this important? From I will say we can have two arguments in favor of the importance of this finding. First of all, from a molecular point of view, we explain, or we try to explain, what is known in the field as functional selectivity. This is a concept that actually started in the G protein coupled receptor field, where they demonstrate that the agonist or antagonist activity of different molecules could be used to select responses, downstream responses. And in case of RTKs, has not been very well described. But what we mean by functional selectivity is actually the fact that two different ligands like FGF7 and FGF10 binding to the same receptor can activate different downstream responses. And besides the nature of these ligands, as an example, FGF7 versus FGF10 regulating different cellular responses, we can also now add receptor localization, so recycling versus degradation, and now also the interaction or the crosstalk with other RTKs in this story, EGF receptor. From a more biomedical perspective, actually localization of RTKs or even the crosstalk between RTKs can actually be used as a novel target in cancer therapy, and we actually found out that 50% of the triple-negative breast cancer from a phosphoproteomics dataset available online show EGF receptor phosphorylation on threonine 693. And in published data from my one former PhD student in Manchester actually show that this threonine 693 phosphorylation is less active, less present in a model of triple-negative breast cancer cell line, which particularly metastasized to the lung. And of course, we are working on these findings. Then we ask another question. What happens when we inhibit FGF receptor recycling? And we use a genetic approach for that. So we expressed dominant negative RAB11. RAB11 is the GTPases which regulate recycling endosomes. So in the absence of RAB11 activity, FGF receptor is stacked in the early endosomes. And then we asked what happened short-term upon FGF10 stimulation or long-term. So starting from short-term, we found out again by phosphoproteomics that there is a big group of proteins phosphorylated upon FGF10 in wild-type cells, which cannot be phosphorylated anymore when the receptor recycling was impaired. And among these proteins, actually, most of these proteins actually localize into vesicles. I show you this picture here because with a PhD student of mine in Manchester developed this nice tool to visualize the localization of group of proteins in contrast to what is available on UniProt, where you can see only the localization of one single entry at a time. And when we look at the gene ontology, we actually found out that this group of proteins were enriching the mTOR signaling pathway. And this was surprising. mTOR has not been described very in-depth downstream FGF receptor to be. So we decided, actually, the postdoc and PhD student in Manchester decided to develop what we call spatially resolved phosphoproteomics to find out whether a recycling-dependent signaling signature exists. And we took the idea from a paper published in Cell in 2017, where they actually fused a biotin ligase called APEX2 to a G protein coupled receptor. And then after giving H2O2 for 30 seconds or 1 minute, they enriched biotinylated proteins through streptavidin-avidin protocol and analyzed by mass spec in comparison of a spatial reference to find out all the proteins in close proximity, 30 nanometers, of the bait, in this case, a G protein coupled receptor. We went a step further, and what we did was actually to fuse Apex to FGF receptor 2b, stimulate cells for 40 minutes with FGF10, because we know that these time points is the time points in which FGF receptor 2b is in the recycling endosomes. And then we collect different samples, I'll tell you in a moment which ones, and we compare the results to the results of obtaining cells expressing RAB11, so the marker of recycling endosomes fused with Apex, or GFP fused with Apex. And of course, RAB11 fused with Apex, in frame with Apex, sorry, is the control for proteins really in proximity of the recycling endosomes. And the GFP fused in frame with Apex actually is the control for proteins in the proximity of the cytoplasm. That for us is actually the background, the noise of our experiment. And what people in the lab, so Michael and Harriet and Joe did in the lab, was to collect not only global proteome and phosphoproteome, but also to enrich for biotinylated proteins in all these samples, followed by the enrichment of phosphorylated peptides, so that we could get what we call a local phosphoproteomic signature. And as you can see here from the principal component analysis, the results of the global and the local phosphoproteome is very different upon FGF10 stimulation for 40 minutes. And we were very happy to see a good number of proteins phosphorylated upon FGF10, both in the cells expressing the receptor in frame with Apex or the recycling marker in frame with Apex, meaning that this is our FGF receptor to be recycling local signaling signature. And among these proteins, very interestingly, we found the activating cytoplasmic tyrosines on FGF receptor 2b, the one that tells us that the receptor is active, and that were found not only in the global approach as expected, but also in the local phosphoproteome, meaning that FGF receptor 2b is actually active in the recycling, on the recycling endosomes. And we also found threonine 693 phosphorylated EGF receptor only on the local, in the local phosphoproteomics data set, confirming what I just told you before, that EGF receptor and FGF receptor are actually active on the recycling endosomes, and they are there together. So when we did the gene ontology enrichment analysis, we found a gain in enrichment for mTOR signaling pathways and autophagy regulators in the local phosphoproteome as compared to the global phosphoproteome. So this was very intriguing for the reason I mentioned before, that the regulation of FGF receptor and mTOR and autophagy has never been really characterized in depth. So I just show you a couple of examples of what we did. So autophagy is that process in the cells that can help cells to survive upon damage. And in cancer can have both a protective but also an oncogenic kind of role. So what we found out in this particular case is that FGF10 actually protects cells from autophagy in wild-type cells, epithelial cells, different epithelial cells. But when we impaired the recycling of FGF receptor 2b by expressing dominant negative RAB11, FGF10 effect is gone. So this means that autophagy is regulated when FGF receptor 2b is on the recycling endosomes. We also found out that this regulation is depending on mTOR activation, because giving mTOR inhibitor here, you see the same result. So the FGF10 protective effect is not seen anymore in different epithelial cell lines expressing endogenous FGF receptor 2b or even an overexpressed version of FGF receptor 2b. And the regulation of this actually happened through the main regulator of autophagy called ALK1, which is a kinase, which is also regulated by mTOR. So at the end, we decide to keep a step further, and we wanted to see what happened in terms of change in protein abundance long-term upon impairment of FGF receptor recycling. These are super preliminary data, not fully analyzed yet, but I just want to tell you that from the analysis of change in the protein abundance, we found out that these autophagy markers, including the kinase ALK1, are actually dysregulated in cells where FGF receptor 2b recycling is impaired by dominant negative RAB11. And we also found an interesting link with glucose metabolism, and we are working, of course, on this. So to conclude this first part and going back to the introductory slide, we can now say that RTK signaling is actually active from all over the place in the cells, at the cell surface, early endosomes, late endosomes, but also now recycling endosomes. And in particular, at the recycling endosomes, cells can regulate a local signaling signature, in this case, an ALK1 mTOR-dependent regulation of autophagy. And on top of that, recycling endosomes are also important to important regulators of the crosstalk between FGF receptor and EGF receptor, and maybe this can be extended to other RTKs as well, to actually fine-tune the balance between cell growth, so cell cycle entry, and cell motility. So overall, this means that the spatial temporal regulation of signaling actually is very important for cell survival. Okay, I now switch to the second part of the talk, where we actually decided to, together with Joe Parson, a PhD student who just finished in Manchester, and a group of Rob Clark, the Manchester Cancer Institute, we tried to understand what happened during breast cancer metastasis. So, as you maybe know, most of the death in breast cancer patients is actually due to the formation of metastasis. And this happens in different organs, mainly the bone, the lung, the liver, and the brain. And this mortality is very difficult to, first of all, predict, prevent, and cure, because once metastases are formed in the different organs, it's very difficult to then treat them in a targeted way, for example. So at the beginning of this project, we decided to compare the proteome and the phosphoproteome of different samples from breast cancer patients, which represent the main molecular subtypes of breast cancer, so the hormone receptor positive, HER2 positive, and triple negative breast cancers. And of these samples, these samples needs to be, frozen. They could not be paraffin embedded. This is a sort of technical limitation of proteomics, phosphoproteomics so far. So this is why the numbers are not very high. But anyway, the key point of this setup is that a clinician helps us to determine the metastatic history of these patients. So we know that 21 of these patients, mixed between the different subtypes, actually metastasize to different organs 5 years post-diagnosis, and 18 of them didn't form any metastasis 5 years post-diagnosis. So our idea was to compare the proteome and phosphoproteome signature of these samples to see if we could prevent or metastasis to a specific organ, or eventually find new targets. And Joe Parsons in the lab developed a very easy method to reduce the complexity of these samples. Obviously, the starting amount of material in this case is in the order of micrograms rather than milligrams, like in the previous project. So the depth of the analysis is not as good, but it is with this protocol, in a very simple way, you can fractionate using a normal plastic way of the lab to go as much in depth as possible before mass spec analysis. Bioinformatics. And of course, what Joe did was also the validation in vitro and in vivo of our findings. Okay, so what we found in the proteome analysis, just to give you an example, was an upregulation of different proteins here in orange. And we focus in particular of this OMD. I'll tell you in a moment what it is. This OMD protein is catch our attention for two reasons. First of all, the abundance of this protein was significantly higher in metastatic versus no metastatic samples. And second of all, the amount of this protein was higher in patients metastasizing to the bones compared to metastasis to all the other sites. So we formulated the hypothesis that this protein called osteomodulin, the expression of this protein called osteomodulin probably correlate with bone metastasis in breast cancer patient. So osteomodulin is a member of the small leucine-rich repeated proteins family, is secreted in the extracellular matrix, and is known to alter collagen structure. This is actually an important link with the breast cancer because breast cancer surrounding, so most of the extracellular matrix is actually composed of collagen, and stiff collagen, stiff matrix are actually correlating with high risk to develop breast cancer. The other known function of osteomodulin is actually to drive osteoblast differentiation. Osteoblast is one of the 2 cell types in the bones. And we know from different in vitro experiments that it can regulate known signaling pathways in cancer cells. On top of that, Joe Parson, actually the PhD student driving this project, found out in the geo data sets that among the third mostly upregulated mRNA present in bone metastasis was actually osteomodulin in comparison with mRNA found in other metastatic sites. And these data sets actually analyze both breast and prostate samples, and both breast and breast samples actually known to metastasize to the bones. So we were intrigued by this finding, and we wanted to understand the function of osteomodulin in driving bone metastasis. First of all, we checked actually whether osteomodulin could be important in the bone itself to allow the environment to attract breast cancer cells, but we didn't find any significant results using different cell models. So I'm not going to present this data to you. But what we actually focus on then is the role of osteomodulin in epithelial cells. And we took a breast cancer cell lines from a triple-negative breast cancer cell lines known and used in the research, which is called MDA-MB231. And we infect the cells with osteomodulin. Of course, osteomodulin is present in the cytoplasm, but also in the conditional medium, as it is a secreted protein. And we tested this function in different assays. We tested, for example, migration. And you can see here by comparing the red bar corresponding to cell overexpressing osteomodulin with a gray bar corresponding to cells, wild-type cells, that the presence of osteomodulin actually increased cell migration. And also when we gave the increasing concentration of osteomodulin in the medium, mimicking, so this is recombinant osteomodulin, which mimics secreted osteomodulin, also the migration of wild-type cells increased. So what Joe did next was actually to inject, intracardially inject cells expressing osteomodulin or not into the mice, and then test after 4 or 5 weeks the presence of epithelial cells, human epithelial cells in the bones by staining for CK19. And as you can see here, again comparing the red and the gray bar, you can see a significant increase of epithelial cells colonizing the bone, the mouse bones, in cells expressing osteomodulin. The green line actually represents the positive control of the experiment, so an MDA-MB231 cell lines produced by the Massage lab in New York, which actually has been selected to go specifically to the bones. So at this point, we know that osteomodulin is overexpressed in epithelial cells in the breast cancer samples that metastasize to the bone, and that it regulates cell migration in vitro and the formation of bone metastasis in vivo. So we asked, we wonder how osteomodulin regulates breast cancer cell motility. So we did a phosphoproteomics experiment, and as you can see here in blue, there are all the phosphorylated sites that were downregulated when cells overexpressed osteomodulin. And in this list, there is the tyrosine 15 of CDK1. CDK1, as I mentioned before, is the regulator of cell cycle. This decrease has been quantified by Western blot, but the key point here to remember is that the tyrosine 15 is an inhibitory site on CDK1. This means that when there is a downregulation of an inhibitory site, it means that CDK1 is more active. So we verified the idea that osteomodulin expression correlates with CDK1 activation by using two published phosphoproteomics data sets that actually show us that in all samples from breast cancer patients that overexpressed osteomodulin here and here, there was a decrease in the inhibitory site on CDK1 here and here. So actually, we thought that if we could inhibit CDK1 with a specific inhibitor, maybe we could rescue the migration of breast cancer cells when presence of osteomodulin. And indeed, this was the case. So CDK1 treatment decreased cell migration in vitro, but also cell viability. You notice here that the simply overexpression of osteomodulin doesn't increase viability of cells per se, but the treatment with the CDK1 inhibitor significantly decreases cell viability in vitro. And just 2 days ago, actually, I got the results that when you inject osteomodulin expressing cells in the mice, as I told you before, in the same experiment I showed you before, and you count how many CK19 positive cells are there, there is a significantly decrease in this number of cells when mice were treated, with a CDK1 inhibitor. And we also verify in an independent cohort of samples from pleural effusions of breast cancer patients that CDK1 inhibitor was important to decrease viability of cells in 4 out of 5 organoids. This was the case. And in particular, in all cases where the viability was decreased, phosphoproteomics actually showed that the CDK1 tyrosine 15 inhibitory site was downregulated. So at the end, we could conclude that osteomodulin could induce activation of CDK1 to regulate cell migration, cell viability in vitro, and the formation of bone metastases in vivo, and that the inhibition of CDK1 decrease the tumorigenic effect of osteomodulin expression. This is important in the context of breast cancer for one important biomedical reason, the fact that breast cancer patients form metastases in the bones, brain, liver, and lungs as primary metastatic sites. But then once metastases, even micrometastases, are formed in the bones, cells can actually migrate to the other site, brain, liver, and lungs, forming secondary metastases. So if we can predict and prevent the formation of bone metastasis, maybe there is a little bit more hope for patients. And of course, the CDK1 osteomodulin axis could be a good target to explore. With this, I would like to thank my team in Manchester. Here you can see people involved in the first part of the project, Harriet and Michael, for the Training 693 story, and that together with Joe, also they work on the local phosphoproteomics story. Jen has been involved in all this project, and Kathy started with Michael the dominant negative RAV11 and glucose link. Paul has been the experimental officer in the lab, working on mass spec sample prep. Jess and Sakim are now working on other projects in the lab. Joe was actually the driving force with Paul and Jen of the second part of the story, so the breast cancer metastasis story, and Hani is a current PhD student in Manchester working on data analysis and bioinformatics of breast cancer metastasis. Here there are all the facilities in Manchester, collaborators, national, international, and now, of course, in Denmark as well, my funding, and finally, a big thank to my mentor, now unfortunately passed away, Professor Ulrich Autenkeller, who attracted me to Denmark and is already really missed. And thanks to him, I'm actually now starting a new adventure at the Danish Technical University. So thank you for your attention.
Chiara, thank you very much. It was a really nice and very interesting talk. So until we get some questions, I'm gonna take the advantage of host. So with regards to the first part of your lecture, so I mean, the mTOR pathway is central to so many different processes. So I was wondering, you studied the contribution of the mTOR for the endosome I guess, recycling pathway in the context of the breast cancer. But do you think that this may be applicable to other types of cancer? I don't know if it's not my expertise. So I don't know if this has been studied in other contexts.
Okay, the link between mTOR and recycling, not at all, because everybody knows that mTOR is actually active on the lysosome. So that is actually the other pathways I mentioned, the one that is needed for degradation. So one possibility here that could be explored in other cancer types as well is that there is a major molecular link between these recycling vesicles unexplored so far that can, and then mTOR could be maybe the coordinator of signaling responses. And as we know, mTOR is also important for metabolism, cell growth, and practically all the phenotype I mentioned today. But the recycling angle has never been explored so far, as far as I know. So it could be interesting to test also in other cancer and also downstream other RTKs.
Yeah, and I guess this could be also applicable in like normal circumstances, not just only cancer, right? So development and it's funny that you mentioned metabolism. So in terms of lipid recycling and that aspect of metabolism, do we know if there's any implication with that? Because there's many dependence with cancer and also development and stem cells. So I was wondering if we can extrapolate.
We know that there is no link between mTOR regulates also lipid metabolism, and we know that there is a change in lipid composition of these vesicles. For example, recycling endosomes are different than lysosomes or early endosomes.
Okay.
Technically, it's difficult to detect exactly what is different. It's something that people are working on. And I will say that if we can demonstrate that this is actually signaling dependent, maybe downstream RTKs, this could really open a new way to explore the treatment. If maybe combine treatment, because we know nowadays that clinicians prefer to have we are going towards a combination of treatment for different aspects that are going wrong in diseases.
Yeah, because you have to look at it from the systems, I guess, approach rather than just an isolated view of the organ or the cancer. So that's very interesting. And I think we had a hand raised during the lecture. I don't know if the person who raised its hand, it still wants to pose a question. So if anybody has questions, please go ahead. But I still have a lot until I don't see anyone else. So with regards to the Apex method that you use, do you think you can gain even more sensitivity using a different approach, or the Apex was the most, I guess, sensitive method that you could use? Like, I don't know, like, can you combine it with electron microscopy to get even more detail into what you're reading?
Yeah, so it depends what you want to do. So the Apex, for example, compared to other biotin-related approaches like the using of the turbo ID or mini turbo, or the old BRDA, allows you to do the experiment quick. So of course, in terms of signaling, we thought that it was the best one, but we wanted to know from a proteomics perspective what happened. You can also validate the findings via microscopy using the Apex, so then to see exactly what happens in the surrounding or the recycling endosomes or other endosomes, and maybe do that in live cells, so you can see over time how maybe the interaction of signaling molecules, but also vesicles itself can work during time. And this then can go back to the mTOR question on to fully demonstrate the interaction or the proximity of mTOR to recycling endosomes. I did that by confocal and it worked quite well, but of course there are, at least for the live cells, we need also then to use more advanced technologies.
And I guess this can, if you go to live, this was in cell culture, right? If I remember correctly, because you have to do the transgenic stuff. So how would you go ahead and look at it into samples, like validate that into that level of detail?
That is good. That is going to be, of course, difficult. And this is the reason why I think that in this case it's better to validate a signature that can be, inhibited in more relevant samples. Inhibition of endocytosis has been suggested in the past, but nothing very specific has been found to block, or to inhibit certain phenotypes in diseases. This doesn't mean that, again, we cannot explore the combinatorial idea. At the same time, we need also to think that these endocytic vesicles are the one that allows the entry of drugs, that allows the entry of medicines. So, and we still don't know enough.
Yeah,
so
exactly.
Actually open other avenues as well.
Yeah, that was going to be my next question. Like, how do you see this going in terms of therapeutic approaches, like hijacking that mechanism
exactly
to heal the system or to help it? So that's that will be very interesting. With this, I mean, mTOR it's like the gatekeeper. So if you manage to tilt it towards your side, it would be wonderful.
And the same about autophagy as well, right? Because again, there is this overview that it can benefit sometimes to block autophagy or not. So it's really depending on the context. And in this case, I think that studying this downstream something that can be modulated, like RTK, maybe it will be important. Of course, in vivo is a little bit more difficult.
Yeah, it's always And in the case of the second part of your talk, where you looked at the role of the RTKs and how they contribute in the metastasis, are you looking to expand that and how if these functions are maintained in the primary metastatic sites and how they differentiate? I don't know if that's
one direction would be to test well how these happens. I mean, what happens in a normal cells counterpart? And of course, if this is also in common with other cancer types like prostate, right? That also metastasize to the bones. At the same time, the idea of trying to predict, metastasis and organotropism has been tried before, but neither genomics nor transcriptomics kind of analysis were conclusive. So this is why we were trying to see, okay, if at least from the signaling protein point of view, we could find something else.
Yeah, definitely. Are you looking at doing like looking at patient samples like from biobanks? Are you trying to do that?
Yeah. We had to do the analysis only on frozen samples. So of course the amount is very slow, and also finding manually linking the clinical history to samples took a lot of time to our clinical collaborators, because GP don't do that, hospital don't do that. The big amount of samples that are paraffin embedded actually will be useful to do TMA, and this is what we are going to do. So try to see if osteomodulin and activation of CDK1 or inhibition of CDK1, depends on which antibodies is going to work, if they correlate with a large cohort of samples with the breast cancer phenotype or response to treatment and all the other parameters that we can think of. This is just the beginning.
Well, you are most welcome to reach out to the Cyprus Biobank. We're always
of course
for new collaborations. And this is my last question, since I see we don't have other questions from the audience. So I'm wondering, have you ever looked at what happens in terms of the neural remodeling in the signaling processes that you study? Because we know that most of our body is innervated, if not all. So have you ever seen or actively researched that aspect of the system?
Let's say indirectly, because during my PhD, I've been studying neural cell adhesion molecule outside the nervous system, in actually pancreatic models and epithelial cells. And neural cell adhesion molecules, so NCAM, modulate actually the trafficking and especially recycling of FGF receptor 2B. So it might be that there could be also a good connection there. Of course, everything is very speculative, what I'm saying, but I will not be surprised that there is also nervous system from the environment that could modulate one of these regulatory mechanisms. Yeah.
Yeah, that's a big, like a hot topic, cancer neuroscience now, like how the nervous system is regulating and it's being regulated or remodeled in this situation. So that would be a very interesting thing to look at. And again, you're most welcome if you're looking for collaboration.
Yeah, of course, we will do that. Yeah.
So I don't want to keep you any longer. As you see, I'm very interested in your topic. We don't have any other questions from the audience. I want to take the chance again and thank you for initiating the sixth season. And we look forward to seeing you in Cyprus also live. So thank you for being with us today.
Thank you.
And enjoy the rest of your day. Continue with this wonderful research. Thank you.
Thank you very much. Thank you, everybody.
Thank you. Have a good afternoon, everybody. Thank you for being here. Bye-bye. Thank you.